Hi everybody, welcome back to Synthetic Biology 1. Today, we've got a math project. I want to get quantitative about gene expression. When you express proteins in cells, you will often want to control how much protein is expressed. Too little protein and nothing happens. Too much protein and the cell can become sick and overwhelmed. But what determines the amount of protein expression that we get? We can answer that question with a simple model. First, let's remember our central dogma. DNA makes RNA, and RNA makes protein. That's what we're modeling, but we aren't going to try to capture every complex detail of this process. That would be impossible, and in fact, it's the complete opposite of what we want. Instead, we want to strip that system down to the absolute bare essentials and produce the simplest model that tells us something useful. Let's start with the statement, DNA makes RNA. Now let's turn that statement into a mathematical expression, in this case a differential equation. We came here to model the production of RNA, so we can write that down first. We'll borrow a convention from chemistry and we'll use these square brackets to indicate concentration. This makes it clear what physical units we're using in our model. When DNA makes RNA, it means that the concentration of mRNA will change over time. So that means we're looking at the derivative of the mRNA concentration. In other words, the rate of mRNA production or degradation. Now we need something to fill in the right-hand side of this equation. What can we say about the rate of mRNA production? Well, in the simplest case, we're just going to make mRNA at a constant rate. That's it. We use the term beta to stand for a constant rate of mRNA production. 1 molecule per cell volume per second, or whatever it happens to be. There's all this biological complexity of mRNA production, but it's just getting lumped into this one constant term. The RNA polymerase binds, and the transcription has to start, and all the little ribonucleotides have to get added and polymerized to make the RNA, and there's all this fussy detail, but no. We're just going to take all of that complexity and roll it into one constant term. Beta has physical units of concentration per time. Now that the mRNA is produced, we have to remember that mRNA doesn't last forever. This is true of most things that you model in biology. A typical mRNA is not super stable, and it only lasts for about 10 minutes before being degraded, digested, and recycled inside the cell. Or mRNA can be diluted just because of cell growth. The cell is constantly growing, which means the cytosol is constantly getting bigger and bigger, and this has the effect of reducing the mRNA concentration. So we'll need another term in our model that we call the degradation term, even though we remember that it also accounts for dilution due to cell growth. That term is alpha times the mRNA concentration. Now, you might ask yourself, why do I have to multiply by the mRNA concentration? Why can't I just use a constant term like we did for beta? Well, there's a few ways that you might think about it. The first way is to imagine that each individual mRNA molecule has the same chance to degrade over time. So the more mRNA molecules we have, the more of them will degrade. Another way that you can think about this is with limit case analysis. Let's imagine that we did use a constant term, alpha, to represent degradation. Well, what happens if beta is larger than alpha? The mRNA concentration increases and increases forever to infinity. And what happens when alpha is larger than beta? Well, then the mRNA concentration decreases to below 0 and then becomes negative. Negative concentration? What even is that? I have no idea. My brain just exploded, and that usually means that we did something wrong with our model. Negative concentrations and infinite concentrations make no sense, and we don't want to use them whenever we can avoid it. Okay, our model is beautiful. Now, let's put it to good use. In our model, mRNA is produced at a constant rate and then degraded at a rate proportional to the mRNA concentration. As more and more mRNA is produced, the mRNA concentration increases until the degradation term balances it out. At this point, the production and the degradation rates are equal, and the concentration no longer changes with time. We call this very important point the steady state. Now, by definition, at the steady state, the mRNA concentration is no longer changing, which means that the derivative term, just by definition, is exactly zero. That means we can do some simple algebra to solve for the steady state mRNA concentration. And we'll find that it's simply beta divided by alpha, the production rate divided by the degradation rate. We can use this model to understand what controls mRNA concentration inside a cell. The production rate beta is affected by many things. For example, just the number of RNA polymerase molecules inside the cell. More RNA polymerase, more beta. We can't necessarily change the abundance of RNA polymerase, but it's nice to see how it's relevant. We do have the power to change beta by changing the promoter for our gene. A stronger promoter sequence will bind more RNA polymerase molecules and therefore increase the production rate. The degradation rate, alpha, is also something that we can change. If we can make our mRNA more stable, then the mRNA degradation rate will decrease. The alpha term also represents dilution due to growth. That means that we can affect alpha simply by changing the growth rate of the cells. Fast-growing cells means more mRNA dilution and therefore less mRNA per cell. These are simple things that you might have guessed even without a model, but the model gives us a quantitative framework to organize our thinking. So, to summarize, first we wrote a differential equation for mRNA production. We simplified the model to the absolute minimum and then we mercilessly squeezed out all of the biological complexity. Then we solved for the steady state mRNA concentration and then we discovered that it's a function of the production rate and the degradation rate. Next, we can go back to the biology to see what tools are available to us as synthetic biologists to change the parameters that matter. But that is a model for another day. Until then, stay productive.
Thank you.